What if I told you that 98% of cancer patients never get access to the most advanced treatments? Not because they don't exist, but because the system is broken. I'm Joelle Kaufman, and today's guest is Dr. Knudsen, CEO of the Parker Institute for Cancer Immunotherapy.
So something that a lot of people don't really realize outside the medical field is that your own immune system is very good at detecting cells that are not you.
Do they do them instead of chemo, in addition to chemo? How does that work?
It entails removing your immune cells, training them outside your body right now to see that cancer and putting it back. Often, though, patients could be cured with a single dose.
Wow.
Yeah, I know. It's expensive, but it's one time.
How do you track your side effects? And you have to track because you don't remember when you show up at the doctor's office what happened 3 weeks ago.
Medicine is an art. Every oncologist will tell you it's not just about the guideline. It really is about treating the patient sitting across from that individual, and that's what makes them so good.
Karen, let's dive in. You talked about the unbelievable change before and after immunotherapy to cancer care. Can you explain, what do you mean? What is that?
Yeah, absolutely. So, when I first got into oncology as a scientist and then also as a healthcare executive leading oncology for one of the largest health systems in the US, so all the science, but all the clinical care diagnosis down, I remember the days before and after immunotherapy very well. Now, for decades, we had surgical intervention, chemotherapy, and radiation therapy as the mainstays of how we treated all cancers. And then in comes immunotherapy. And the first in was really identifying new ways to train your own immune system to see your cancer. So something that a lot of people don't really realize outside the medical field is that your own immune system is very good at detecting cells that are not you. People often understand that that's the case if you're detecting bacteria or a cell that's been infected by a virus, something that it should eradicate from existence. It also recognizes cancer cells that were you and still are, but have mutated to the point that they've attained cancer characteristics, and your immune system is trained to see it. So that's happening all the time. It's happening right now while you and I are talking. And what immunotherapy, or a class of immunotherapy called checkpoint inhibitor therapy, is designed to do is to eliminate a cloaking mechanism that tumors use to hide from your own immune system. And so checkpoint inhibitor therapy essentially allows the tumor to be seen by your own immune system. And it's been an incredibly powerful anti-cancer agent for cancers that were very difficult to treat. Aggressive lung cancers, metastatic melanoma, which, really there were so few potential therapies to use against, and bladder cancer. And then the list of cancers continues, initially starting off as just checkpoint inhibitor on its own, and now in combinations, smart combinations based on science with immunotherapy, radiation therapy, and other types of immunotherapies as well. So it's opened up a whole new door for us. And that's just one class of immunotherapy.
Help us, what is an example, maybe a couple of examples of the drugs that are these immunotherapy agents? And do they do them instead of chemo, in addition to chemo? How does that work?
All the above is true. So let me zoom out for just a quick second and talk about drug development and drug delivery. The way that it works is if you have developed something that you think is a new curative therapy, this is our goal at the Parker Institute, it's what we do very well, then where that gets introduced, once it's gone through preclinical testing to anticipate toxicity, to anticipate how well it would work and what potential challenges might look like. Then you enter into the phase of clinical testing. And the way that works in the cancer space is that it gets added on at the very end of when someone has exhausted all the options that are what we call standard of care, meaning that there's good evidence that therapy A, B, C, and D have a potential to cure this patient or knock back its disease and give that patient a great quality of life. Once a patient has gone through those A, B, C, D, the standard of care, and fewer options are left, is often where new potential curative therapies enter the market. It can also enter into a space where the new potential curative therapy is given in combination with standard of care. And a trial would be run so that you and I both had standard of care, but we would be randomized into one of us also receiving the new agent. So this is where immunotherapy started, with these very challenging, difficult to treat cancers like metastatic melanoma, for which patients had run through options, or aggressive lung cancers where patients had run through options. And it started to show benefit, that these patients were living longer, their tumors were knocked back from these cancers. So when that happens, then drugs can move what we call earlier in disease course, now with an earlier standard of care, or maybe at the very beginning, once you start standard of care. And every cancer is considered differently, and as appropriately so. We're still finding the space for immunotherapy. What are the right combinations? When's the right time to intervene? But they're moving earlier in disease space, in disease course, which is an indication that they are really promising in a large number of cancer types. Now, that's the checkpoint inhibitors. But then especially in the hematologic malignancy side, let's call them leukemias and lymphomas, we have an entirely different class of immunotherapies called cell-based therapies. And that's a slightly different strategy. The strategy there is not to use drugs like checkpoint inhibitors to allow the tumor to see your cancer. But this one is really quite bespoke and targeted. It entails removing your immune cells, training them outside your body right now to see that cancer and putting it back. And those types of therapies, these cell-based therapies, have worked wonders in a large number of leukemias, lymphomas, multiple myeloma, which, over the course of my lifetime went from being truly one of the most difficult cancers to treat to now having so many options that we need to think about quality of life for that patient in terms of deciding which one to go for. So we know that cell-based therapy can be curative. It's intensive, as described, to remove someone's immune cells, train them, and put them back. Often, though, patients could be cured with a single dose. So,
Wow.
this,
Wow.
yeah, I know. It's expensive, but it's one time. And so how do we take that, which is working so well in leukemias and lymphomas, and move it into what we would call the solid tumor space, the things that don't come from blood cells, things like breast cancer, prostate cancer, bladder cancer, colorectal cancer. So that's where so much of the science is moving right now. And I think we're going to see vast breakthroughs over even just the next 5 years based on the science that we see coming up.
Not
If I
just
understood it right, hold on. What we've been doing in the hematological cancers, and this isn't a bone marrow transplant, this is something else. We've been taking the immune system, your own immune system, retraining it, and it's going, and that now it knows how to catch the deceivers,
Yes.
and it goes and it does its job. And we want to take that very personalized approach and figure out how to do it at scale and with efficacy for the solid tumors.
That's exactly right. Which are traditionally much more difficult to treat in the metastatic setting. And if you think about it, why would we go to the trouble to develop something that's so technologically challenging as to remove someone's immune system, train it, put it back? Because it works. But also it's the case that if you think about it, it's there a lot longer. So if you're taking chemotherapy, that chemotherapy is working during the time that it's coursing through your veins. If you have radiation, radiation is working during the time that you're under the beam or with the radiation agent implanted. Your immune system, once trained appropriately, is there beyond just that initial time. It's with you. It still remains your immune system. It's Karen, it's Joelle, it's who we are. And it's the new immune system for us that's now trained to see these cancers. So the benefit is really obvious. And the question is how to scale it, you mentioned. So this ability to remove someone's immune cells, train them and put them back is, of course, quite specialized. And there's risk associated with it. So how do we reduce the risk and make it much more accessible to other types of cancers, and to make it so that more patients could take advantage of cell-based therapy? There are emerging technologies that we call in vivo, which means we don't need to take out your immune system to train it, but we can use new technologies to train your immune system while it's still in you. So there are multiple different strategies right now that are in clinical testing for training someone's immune system, which is called this in vivo technology, means, just in you while it's still in a living, breathing person. And I think we're going to see some winners relatively soon in that in vivo technology space. It's a race to what technology can work the best.
Is that different? I mean, the checkpoint inhibitors are infused during an infusion and, they interact with your immune system and off you go. How is this in vivo different from an infused
Yeah.
medication?
Great question. And it would be infused. And it's a way of infecting your cancer cells or infecting your immune cells rather, so that they can actually be retrained in the same way that happens in a laboratory right now for cell-based therapy when they're taken out, but to allow it to happen within you using advanced genetic tools where we can do some manipulation of your own immune cells, but without having to take them out. So this allows us to skip a lot of the steps. But the biologic basis is different than immune checkpoint inhibitors, where immune checkpoint inhibitors essentially disrupt that cloaking mechanism that's specific to the cancer. In the cell-based therapies, you're actually training the cells of your immune system directly and modifying those directly. So it's a slightly different strategy.
Sure. Sure. Okay. So it sounds a little like I was reading about this triple negative breast cancer vaccine that it literally just did stage 1 trial. So it's, I think, 36 people, not, and it proved that it was safe for 36 people. And they're looking at stage 2. But it sounded a lot like an in vivo immunotherapy approach because to some extent, aren't vaccines kind of similar to this concept?
So I'm going to put vaccines in a third category. So the way vaccines work, so checkpoint inhibitors are disrupting that cloaking mechanism.
Right.
Cell-based therapies are changing your own immune cells directly so that they're smarter. They can see the cancer and are going to be attracted to the cancer that we know you have. Vaccines have a same goal, training your immune system to see a cancer, but it's through things that we know already work. So the way the vaccines work, if your vaccination against really anything that you want to name, whether it's childhood vaccines, vaccination against the human papillomavirus, the way that works is by training your immune system or hypersensitizing your immune system to see proteins that are presented to the immune system. And that can occur by those proteins decorating the cell of a cancer. So there's been a whole discipline of biology that's been looking at triple negative breast cancer, for example. What are the proteins that decorate the outside of a triple negative breast cancer cell that's in Karen that's different than a normal breast cell in Karen? So the way the vaccination technology really works is by looking at what are these new proteins that are specific to the cancer that are decorating it, and then using that in a standard vaccination strategy to train your immune system to see it. And as we all know, vaccines can be for life, with boosters sometimes, but for life, where your immune system won't forget that this is the kind of thing that should be destroyed. So vaccination I'm very excited about where we are going with vaccine technology because there's some commonality in proteins that decorate the surface of cancer cells between individuals, amongst individuals, where off-the-shelf vaccines can be developed. There was a really fantastic study as well or earlier this year. It was a smaller study, even just with 9 patients, looking at patients who were with a solid tumor that were destined to go on, or scheduled rather, to go into surgery. And while they were waiting to get to the time of surgery, they actually were given a vaccine to see if they could shrink the tumor. And some of those patients had tumors that shrank to the degree that they actually no longer needed surgery at the end.
Wow. Wow.
Showing new paradigm in oncology that you could cure without cutting. We know that surgical removal of a cancer, if it hasn't spread, is often a really, effective way to cure cancers. Look at diseases like prostate cancer. You have, if it's truly just confined to the prostate, upwards of a 99% cure rate if you can take it all out. If there's a way to have your immune system do that and skip some of the negative repercussions of surgical intervention, then that's a win for patients. So this is something we are investing in heavily at the Parker Institute, and encouraging others to come along with us, because we believe that vaccines are a great way to scale immunotherapy so that more patients can take advantage of immune activation.
So, what I've learned, and really appreciate you teaching me and my listeners, we have checkpoint cloaking immunotherapies that are teaching the immune system a new trick.
Yep.
Great. Then we have, I'll call it a systems upgrade, the in vivo, where we are upgrading the immune system to know how to fight and destroy these kinds of cancer cells. And it's, again, very bespoke. And then we have the third, which is the protein hunters, right? We're training the immune system to look for certain proteins. And as you were saying that, the triple negative breast cancer one I was reading about last week was using an a lactation protein that only shows up with you're lactating or you have breast cancer, triple negative breast cancer. So, so it's really interesting. We've got these three. Now, people are worried with immunotherapy. There's a lot of buzz about vaccines, whatever, about long-term or even short-term side effects. At your institute, I'm sure that's part of what you're looking at. What can you tell me about the risks of this type of therapy?
Yeah. So, the risks start to become emergent even before someone actually receives the very first immunotherapy, or a new immunotherapy that's been developed, because we can mitigate some of those risks and anticipate them in preclinical models. So, models in the laboratory that include everything from animal models to actually human tissue models can start to become increasingly instructive as to what type of potential repercussions there might be. And you, of course, learn much more in the clinical testing phase. As you talked about for that triple negative breast cancer study, it was to look at safety. And it was I believe that one met the primary endpoint.
Did.
Goal.
Did.
And so now it has an opportunity to go on to determine whether or not it actually was able to meet the goal of shrinking tumors, even though I'm sure that they were measuring that in that study.
Mm-hmm.
The first pass is always safety. So safety first is the way that we do drug development in this and other modern countries. So that's how. Now, we are still learning about the overall survivorship impact of someone who's a long-term survivor of cancer after having had a checkpoint inhibitor or a cell-based therapy. And certainly vaccines are in their very new phase for us in the oncology world. So we're still learning. And what I would say is, these are patients that actually just overall, we don't know a lot about what it's like to be a long-term survivor of metastatic melanoma. Because frankly, those patients were few and far between before immunotherapy. So it's even beyond just what are the negative repercussions of immunotherapy, because often it's given after, as we talked about, that's care of radiation and chemotherapy. So it's that entire combination of the suite of therapies that a cancer patient has been given. And what are the long-term effects? That's frankly the phase that we are in now. But what I would say is that we're buoyed by the fact that these long-term survivors are actually in general doing quite well. As we learn about side effects that happen, of course, attempts to mitigate them will happen in the clinical trial space as well. So we're happy to be in that phase where survivorship is something we're thinking about.
Yeah, well, I'm a very appreciative recipient. So let's dig down. You mentioned the clinical trials, and we talked about what you're doing, these other scientific approaches, and they are amazing options for many patients. But you talked with me previously about how hard they are to access, whether it's the location, the travel, the matching process, and that you're using AI, agentic AI, to help with this. Can you tell us a little more about what you're doing to try to solve this problem?
Yeah. So, it's been longstanding a problem. I'm 55 years old, and in my entire career, as a scientist, as a healthcare executive, as a CEO of the American Cancer Society, now the CEO of the Parker Institute, in every role I've ever held, I've been part of that community trying to get more patients access to clinical trial. As a healthcare executive, I was very proud to have put clinical trials out in the community, which is not simple to do unless you're a large healthcare system. And it was only when we really achieved scale that I was able to do that. And I was very proud of that endeavor. But that's one geography. How do we scale that? At present, in the United States, we can still count on less than two hands the percentage of patients that go on to clinical trial. And frankly, most patients are not offered access to a clinical trial, which in cancer truly is the most advanced form of care. As we talked about, that's the place where you get standard of care and likely a new therapeutic that is intended to improve and extend your life. So how does that happen? I can tell you from being a healthcare executive that the bottlenecks to getting patients to care are profound, to clinical trial, even when everyone is motivated toward that end. If you think about the academic healthcare system, people work there and in the academic center, often making less money than they would out in the community because they really believe that clinical trials are a core component of cancer care and they want to give their patients every possible option. Patients go to the academic centers because they want those options. But the manpower that's required to ask questions like, should we actually open this trial at our site? Do we have the right patient population? Do we have a trial that's competing with one that we're proposing to open? So in which case you wouldn't meet your enrollment goals. Would you have enough patients to really answer the questions that you're trying to ask? Which was the right one for your patient population? That's one suite of questions. The second is, how do you match them? For every clinical trial, there's a list of inclusion and exclusion criteria. Inclusion must be true. Patient must have this stage of disease. Patient must have been on 2 prior lines of therapy. Patient must have fill in the blank metrics associated with their own, health. And then what can't be true? They can't have ever been on this drug. They can't have ever been on or shown that they have this type of side effect. So that the list is long. The manpower required for assessing any individual patient on those inclusion exclusion criteria is hard. And then there's the third tranche of third bottleneck, and that's pulling down the data. So patients who are on clinical trials get extraordinarily attentive care. So even the standard of care arm, there's much more assessment of that patient, of making sure that we truly understand every sniffle, every itch, every rash, so that we can really, watch them very, very closely since they have entered into this clinical trial. So the data required to pull down and collect that and assess how it's looking for every patient is a lot. So it's a lot. Should we open the trial? How do we match the patient and how do we pull down the data? It's a lot of people. Now go how clinical trials are funded. Every clinical trial is generally underfunded. It's very difficult as a cancer center director and healthcare executive to piece all that together and hire those people that you need to do it well. And no one wants to open a clinical trial and enroll a patient unless you're certain that it's the right trial for the right patient and that you can do it safely and address the needs of that individual. So Now we're in this great space, 2025, where artificial intelligence and agentic AI is helping us. So, separate than the Parker Institute, I happen to be on the board of a number of privately held and a publicly traded company. One of the privately held companies is Paradigm Health, really incredible group. Where, Paradigm actually is using agentic AI to solve for all three of those bottlenecks and meeting their goals of already helping community health systems open clinical trials, because for the first time, they don't need to hire a whole suite of people. They need to hire some, but not nearly as many. And to have those specialists in clinical trials in the form of agentic AI assisting them out in the community now is really truly starting to make it so that there's more equitable access to clinical trial. I feel very bullish about the fact that companies like Paradigm and others are going to help us truly close that gap, get clinical trials out into rural America, and make it so that patients aren't having to travel long distances to go for care, or choosing not to do that and choosing less optimal care because it's closer to home.
I think that's amazing. That's one of the really positive things that can come out of this whole AI revolution. There are so many. But you talked about new technologies, new companies. And one of the things that's very hot right now is this whole longevity space, survivorship, longevity, and people saying, oh, should I get a full body scan? Should I get a Gallery blood test? And, there's actually a lot of debate about that. And, is that effective for, risk management? So what
Yeah.
do you think?
Yeah. So this is such an important question. And I think one that if we can illuminate for people, they can start to take agency of making choices for themselves. And I say this, as someone who's been in oncology my whole career, but also as an employer, as a CEO now for two different organizations, I've needed to think about, what are the things that are important for us to offer and our own HR benefit for people. And the dynamic that's playing out now is that we've had cancer screening for decades. And cancer screening is by definition for individuals who are of average risk for cancer. But I've had a little fun I don't know if you're going to call it fun, but I've done my own set of experiments over the last several years of when I'm in a room of people, even if it's thousands, asking how many people are of average risk for cancer? Raise your hand. Almost never does anyone raise their hand because they just don't know. So we've put quite a lot of burden on primary care providers and on patients to make their own determination about what their relative cancer risk is. So we're entering into this era again with technology where risk will start to become fine adjusted. How do you know if you're of average risk? It's more of absence of things, absence of a known genetic risk, absence of a personal history that's going to increase your risk, and the absence of a familial risk that might be meaningful. Now, most individuals, even in healthcare, aren't so equipped at being able to adjudicate that for themselves. So, how do we make it easier? Well, tools to assess family history, to document so that in a perfect world, every patient would get asked, what is your family history of cancer every year when they're at primary care? And primary care has tools that are enabled to help them understand if that person has a familial history that should change their cancer screening. Part one. Part two, genetic risk. We're in the spit in a tube era where it's very easy for someone to spit in a tube with organizations like Color Health, which we used when I was in healthcare, and I had a partnership with them in healthcare as well, and to understand what your genetic risk is. This is the genetics that you're born with. You only have to do the test once, ever. And what that tells you is whether or not you're carrying a genetic alteration that changes your risk profile. The thing that I think people would be most familiar with is a BRCA1 or BRCA2 mutation. Okay, well, then there you go. So, and not all mutations are created the same. So there are BRCA1 mutations that are not so meaningful for cancer risk and others that are really powerful.
I didn't know that.
Risk changes.
I didn't know that. And by the way, for the listeners, I waved my hand because I'm a BRCA1 that was very known to be very high correlation with triple negative breast cancer.
Mhm. So, and same for BRCA2. These are big genes. Not all mutations are created equal. So today, if I, and I have to say, I've done that. I've spit in the tube. I know my genetic risk of cancer as it stands today. Now, I only need to do the test once, but the understanding of the science will continue changing. So some of those mutations in BRCA1 or BRCA2 that right now we're not really sure, are they related to cancer or not? Because we haven't studied enough people. They have a designation called VUS, variants of unknown significance. Well, the more that science plays out, the more these VUS mutations get binned into meaningful for cancer, not meaningful for cancer. So I'm very grateful that I have out there where I spit in the tube and they did my genetics, a group who's going to contact me. If the genetic information about me, now we know more about, and there is a change in cancer risk, I'll modify my cancer profile. So I don't have a genetic risk of cancer. I have some familial risk of cancer, but I have a personal history that increases my cancer risk. So I have a different screening program than most people because I'm not of average. I'm one of those people who knows I am not of average risk. So for me, I'm very attentive to that, and I'm grateful to have a primary care team and oncology team around me that helps me constantly set and monitor my screening risk. So what I'm experiencing, I think, will be in the very near future, the ideal for everyone. So understanding your cancer risk, using that information to utilize behavioral change, and understanding that 40% of cancers are tied to a modifiable risk factor. So how do you modify your risk if you know you're at higher risk? How do you mitigate against that? That's something within your own control. And then having your own screening program that's bespoke for you. Now, coming up, and you mentioned these, are blood-based tests to determine whether or not you've got a cancer that's there, but we can't see it yet through traditional screening. Remembering that most traditional screening, not all these days, but most traditional screening that's approved is based on imaging. Can we see a breast cancer? Can we see a prostate cancer? Can we see a colorectal cancer? All of these things are true. However, what if there was a world where you could do a blood draw and see whether or not someone had cancer DNA circulating within them? And that's the era where we are right now. They're called MCEDs, or multi-cancer early detection tests. So what these tests are designed to do is to see DNA that's evidenced of a cancer that came from a cancer that's in your circulation. The same type of technology is used to do genetic testing for pregnant women to determine whether or not the child that they're carrying or the embryo that they're carrying has genetic alteration, so that they can plan accordingly. That same technology is now being used to look for cancer. Can it see cancer DNA? Yes, the data on that are very clear. What yet has to happen is to show that they have clinical benefit. So I know that might sound confusing, because we all know if you treat a cancer early, you have a better chance of surviving. But what do you do with the woman who had a blood test and it says, looks like she may have breast cancer, but her mammogram, even an intensive diagnostic mammogram, isn't showing anything, and an ultrasound isn't showing anything? So what do you do with her? How do you counsel her? And does the blood test detection really mean that she's going to actually develop a meaningful breast cancer? That's the kind of study that's still undergoing right now. Because as we talked about before, your immune system is extraordinarily good at seeing cancer and destroying it. So just the fact that you had circulating DNA of a cancer within your bloodstream doesn't mean that it's an active, clinically meaningful tumor. And so that's where we are right now in this state of play. So studies are ongoing to determine whether or not these blood-based tests are going to be helpful in helping people do the things that they really care about, detect cancers that are meaningful early, and if they do get cancer, to survive that cancer because it was detected early. So the data are playing out. Where they're also being tested, which I think is incredibly useful and promising, is in someone who had cancer, who was given what we would call definitive treatment, what we thought was curative treatment. Let's take the prostate cancer guy. The guy who I said had a 99% chance at cure if it was truly localized to the prostate and he undergoes an effective surgical extraction of the cancer. So then how do we, can we continue to watch him? We do that right now by looking for a protein in the blood called PSA, which is tied to prostate cancer. But is there a world where we also look for DNA return? It looks like their prostate cancer DNA return. So looking for whether or not someone is going to have a recurrence, I think is where there's significant amount of clinical testing right now that may be more rapidly applicable. But there's a long way of answering your question to say the jury's still out as to whether or not these types of interventions that are not the standard of care testing for screening bring clinical benefit.
Well, and I think it's interesting, this understanding your risk. Over at UCSF, Dr. Laura Esserman, I think, just released some findings from the Wisdom trial, which was 40,000 women enrolled, where they saw that doing the genetic screening and doing risk-adjusted screening was as effective, I believe, as broad-based, I'll call it smushing, mammograms. And the fastest growing group, percentage-wise, are women that don't even qualify for mammograms because they're too young.
That's right. And
right. So,
and
go ahead.
It's the U.S. public on this, right? Because screening is for women of average risk. But what the American Cancer Society says the screening guidelines should be is different than what the U.S. Prevention Services Task Force says it should be. So women and primary care are caught in the middle, like what to follow, what to choose. And so, I do think that this risk-adjusted screening era, it's just coming. And, we have so much potential information about ourselves now, the genetic risk, the familial risk, the known personal history, which could change your risk. These are going to impact what we do, even if they're not very specific screening guidelines. Knowing that information and having your primary care intake that information may change the way that they respond to a symptom. Let me give you an example. So the number one cause of cancer death in the United States, and then by far for men and women, is lung cancer. So how do you get screened for lung cancer? The screening guidelines for lung cancer are for people who are smokers. So this is unlike the breast cancer, prostate cancer guidelines of average risk. Lung cancer is this exception where the only way that you get coverage to get a low dose CT scan, a low dose CAT scan, is if you've been a significant smoker over a period of time, 20-pack years. Okay, important and key for those people to get screened and followed.
Sure.
Really important. But what's the number 2 cause of lung cancer? It's radon. How many people know if they have been exposed to radon? Well, if you're, so obviously people with a basement, have you tested your basement for radon? You can't just test it once. It's actually something that goes up usually in the winter months. So unless you've got a constant, if you have a basement and you don't have a constant radon screen, how do you know? Someone who lives in a house with a well, which a lot of people do in rural America, radon, it's naturally occurring. It may be in one house and not in the house next door, but it comes in through the water and becomes aerosolized. So every time you're taking a shower, you're breathing in radon. So how do you know? Well, if you know that you've been exposed to radon because you've tested it, you've realized, oh my gosh, I've been exposed to radon, I've increased my lung cancer risk, understanding that's the number 2 cause right behind smoking, then now this person's symptoms may need to be taken in a different light once their primary care knows that they have had a health exposure that's modified their risk, that's different than a familial or a genetic risk. And increasingly, that understanding, I think, will also modify how we are viewed as individuals, so that we actually are being viewed as individuals and not large categories of people when our cancer risk assessment is going on.
It totally makes sense. Now, another thing I think that makes risk assessment and ongoing management really challenging is what you called the 32 provider problem.
Yeah.
right? So, cancer is multidisciplinary, oncology is multidisciplinary. Uh, the person who sees everybody and knows everything is the least educated and knowledgeable in the whole mix because it's me, it's the patient. So, how are you, and by the way, immunotherapy and AI and all these cool things, how are we going to change that experience? And I think you mentioned something called a patient agent.
Yeah. So, it is really, you hit the nail on the head. The average cancer patient now in the United States sees 32 different providers during their journey as a cancer patient. So, if you just imagine trying to make an appointment with the same hair salon 32 times, that's already miserable. So now, how do you manage this as a patient to really understand how to navigate what's next and who is going to be your quarterback in this? So, it's not just confusing for the patient, it's also confusing for their caregiver. Almost invariably, patients have a caregiver that's someone within their family sphere. And I'm going to use family and friend here, together, who are helping them through this complex journey. As the cancer patient is really focused on trying to be well, they need someone to help them navigate. Now, there are navigation strategies that can help with appointments and that can help with, um, that can essentially help with the logistics and helping you find a ride. And those are coming out. But I did co-found a company with a really phenomenal individual, Justin Green, called Patient Agent. And it is intended to solve for the understanding components, relying on, we're still in stealth mode at the moment, but working with health systems to pilot our concepts of ensuring that we demystify for patients information about cancer, about every aspect of that patient journey, what to expect on different types of therapeutics, questions about their own individual treatment plan by connecting into their own health record as the patient, and as well pushing notes over to the care team so that when they're going to their oncology visit, it's a more informed visit. And much like the physician writes notes for the patient when they leave and it's there in their health record, the reverse can also be true of the patient and their caregiver generating a note to say, here's what we've seen in the last 4 weeks. Here are the questions we have and the things that really concern us. So when the door opens to that visit, everybody already knows what's happened. So we're in this very exciting pilot testing phase here where we have engaged patients, caregivers, oncology teams, but also oncology social workers and patient navigators to solve for those gaps in understanding. The challenge with cancer, well, it's 200
So
different
many.
diseases, right? But the way that you treat, as unfortunately had to learn, a triple negative breast cancer is very different than an estrogen receptor positive breast
Mhm.
cancer. But even in those journeys of what to expect, we know what standard of care in general looks like, step one, step two, step three. But in between each of those steps is a scan, is lab testing that can change step one, step two, step three to a completely different journey. And so, how it is that patients and caregivers can be supported in those deviations from plan because something wonderful or something not wonderful has been observed is part of what this is about, with the full belief that educating patients and caregivers about their journey and giving them a tireless 24/7 agent to talk to that's acting on the patient's behalf is going to improve outcomes.
Well, I think that makes sense. It's part of why I ended up writing my book and why I do this podcast is my approach to being that quarterback and my familiarity with the starts and stops and the rhythm of the journey was far beyond most cancer patients because I'd seen this movie, earlier versions, but I'd seen the movie and I'd seen it 12 times. So my ability to be a partner and to be informed and to then create tools, you're talking about coming in prepared. I feel like I'm being disintermediated in real time, which is great. I'm thrilled. But, I created and I share digital spreadsheets.
Yeah.
How do you track your side effects? And you have to track because you don't remember when you show up at the doctor's office what happened 3 weeks ago. And even if the doctor knows to ask, it's not a meaningful conversation, which, by the way, back to your point on taking a family history, there are whole cultures in the United States and probably outside of it where people don't talk about what killed Aunt so and so, grandpa, or what have you. It's considered, bringing the evil eye, but they don't know. Like my particular subculture, we talk about it. And, my family, we've been talking about it with our kids since they were small because we knew we had this genetic predisposition. And so better that they know. I think what you're doing with Patient Agent, I hope it works. And I hope it helps patients get better care and providers give better care. I mean, that's the idea. So you've been awesome. I wanted to ask about something you brought up when I heard you at CCX around the innovation bottleneck. Like it sounds like there's so much going on and it really sounds great, but you said, but there is actually a bottleneck and it's this tension between investors and the National Institute of Health and how we get to breakthrough. So can you explain what the innovation bottleneck is and what people can do about it?
Yeah, absolutely. And it's something that I think is getting worse over time. We're accumulating all of these on the science side, really promising strategies, not just for therapies, but for prevention and early detection. And they're piling up in laboratories. So how do we get them into the phase of clinical testing and into commercialization, which is how people get access to new science that can be life-saving? The world has changed. So, it used to be that there were venture capital groups who would come in, if a great idea, let's form a new company, and then we will help to support that clinical trial. And if it starts to look promising, then we take the next step to determine how we get into even the bigger trial. And ultimately you get approvals for, the life-saving therapies that we've had in the cancer space. That was previous days. Where we are now is that everyone wants science to be what we call de-risked, which means it's not just a great idea that was in the lab, but show me a clinical signal. Show me that something is likely to work. That's where we come in at the Parker Institute for Cancer Immunotherapy. And we are this simple and radical model of one. I hope and I'm here to grow it, but we need more of these types of models. So here's really what we do. We get behind game-changing big bets. We will not fund anything less than high-risk, ambitious science geared toward cancer cures. That's our thesis. We're relentlessly focused on that. Okay, so we do fund research, and that's an important component. But increasingly, I think that the secret sauce of what we do lies in the next phase, which is that we don't just fund it and forget it. Fund this great science that's happening in someone's laboratory. But we actually put someone in the technology transfer office so that if I'm funded by the Parker Institute, that person that's in the technology transfer office doesn't just see my discoveries, but sees how my discoveries relate to the other 163 scientists that we have funded right now doing ambitious research. So then that results in our ability to say, you know what, this plus this equals 12. And so we're going to put that together in the form of a patent or in the form of a new company. We're a not-for-profit that gives birth to for-profits. So we have 17 companies in our portfolio where we are the lead investor or part of a syndicate. And when those companies hit, or the technologies, if we license them, hit, that comes back in so we can fund more science. No one's getting rich off this. This is all about getting innovation to people. And so it's a really unique model where venture capital has seen what we do because we don't find our founders in the wild. No one's pitching us science to get behind. We've seen it from the very beginning. And so As we then get behind it, then venture capital comes along. And in our 17 companies, a little more than $4 billion has been raised. And so that's allowed our companies to get to those phases of clinical testing, get the signal that allows other investors to want to continue to come in and hopefully generate the next wave of cures. So it's a very different model that is intended to do one thing alone, and that is to eliminate those bottlenecks so that we're shortening the time from a great discovery, getting into clinical trial and getting into more patients having access to curative therapy.
Of course, the other company you're involved with the agentic AI to make the clinical trials more efficient, the patient agent to help explain and keep the patient involved. It's a fabulous ecosystem, Karen.
Thank you.
You know, as someone who I say, cancer is my family's thing, I'd like it not to be. I'd like it not to be. But if it's going to be my thing, I want us to obliterate it. I want it to be a speed bump, which is what I consider my personal journey was. Very thankful for that. But this was terrific. You have been a fabulous guest. And I just want to ask my closing question. What's your advice to people who are dealing with cancer on how they can kick cancer's ass?
Yeah, I would say ask questions, always take ownership. And every time that you are in the oncologist's office, ask those questions about why this therapy? What are my alternatives? What else can I expect? Just take ownership over that journey as much as you possibly can if you've gotten a cancer diagnosis, and be a relentless seeker of information. And I have to say the best care teams revel in that environment and love the patient who really wants to push the ideas and really think through what is the right plan for that patient. Because medicine is an art. Every oncologist will tell you it's not just about the guideline that says, here's the next right thing for this patient. It really is about treating the patient sitting across from that individual. And that's what makes them so good.
Terrific. That's great advice. Thank you, Karen, and much, much success to the Parker Immunotherapy Institute and everything you're doing.
Thank you. I think you and I have the same goal. Love the idea of kicking cancer's ass. It's what I get up every day to do.
That's awesome. Great.